Okay, can somebody confirm you can hear me?
I can hear you.
Great, thank you. Before we begin, are there any questions? Not hearing anything? All right, let me talk a little bit about the grading. I know I got Lab 2 graded, but I got it graded, well, actually early this morning. So it's not going to be due this Saturday. I postponed it until next Saturday. Any questions about that? So lab two, if you didn't get 100%, you can resubmit it, not by this Saturday, but a week from this Saturday. And then it'll be graded a second time. I need to look and see how students voted. I haven't done that yet, but let me get to the grade book because I graded a few other things. I know I got the extra credit picture graded, so you got a grade in there. Need to get the extra credit study 2 graded. I got worksheet lecture 2 graded. I think that's about it. I have not graded the quiz 1C, or I think the practice quiz 1B. So I need to grade the question on did you show me your environment? I have graded the retake of the environmental quiz, but I've not moved your score to the environmental quiz. So I need to do that. And once I move it, the retake environmental quiz will not count in the grade book. You will still be able to access it, but the environmental quiz does count. So there will be 1 point of credit on that one. And where is that showing here? Yeah, it's out of one. The retake currently is out of one, but I'm going to move it so that it will not count in the grade book, and it will not even show in the grade book when I remove it from not counting in the grade book. But you can still access it by the link that I put on the web page. Retake the environmental quiz. Any question about that? Generally, students who took the retake did better. Boy, I don't understand why. I'm just looking at one student here, and uh, she got 100% of both the environmental quiz and the retake of the environmental quiz. I don't recommend doing that because if you didn't get 100% on the retake, you would get a 0 on the retake, and then your environmental quiz would have been changed to a 0. But most students did better on the retake. There were a few students who still have not shown me their complete environment, and they got a 0 on the retake. I think there's only one, but all right. Any questions about the grading? I think that's it. On the grading, let me shut down the grade book and then I'll show you my screen. Well, we're not going to go there. All right, should be seeing my screen. Today is October 9th, and we should be doing Lab 4 stains. Any questions about what we're doing? Any questions about anything in the future? I need to upload this. Maybe if I leave it here, I'll remember to upload it.
I do have a question about the future, I guess.
Mhm.
Um, how are there going to be any indications on what is on the quiz? I thought the quiz that we had last night was going to be chapters 1, 2, and 3 because we had already been in chapter 3. And that we were also going to have access to the PowerPoint that you were using in your lectures for chapters 1 and 2.
Let me share my screen here. I don't think I'm sharing it. No, come on, let's get here. No, I'm sharing it, right? You can see my screen?
Yes.
Okay. So, quiz one, it states on the syllabus, and I thought I stated in the lecture, is going to cover chapter 1 and 10, and only the start of chapter 2. It doesn't even include the last part of chapter 2. And that's because we're still covering chapter 2. And then labs 0, 1, and 2. So that was quiz 1. Any question about that? Anyways, what
Thank
is
you.
on the quiz is always on the syllabus. And generally, I do state it when I talk about the quiz. Quiz 2 is going to cover chapter 3 and the end of chapter 2, as well as labs 3 and 4.
Thank you.
All right. I want to keep this here, and then hopefully it'll remind me to upload that because I changed the date of quiz 2C. I've got a link for 2C. It's actually right there, quiz 2C. All right, if there's no further questions, let's go on to lab 4. That's the exercises. Lab 4, staining techniques. We're going to talk about the simple stain, the differential stain, and special stains. As in all labs, there is some reading that you should do before you begin the lab. So you should read about the size, the shape, and the arrangement of bacterial cells. Only 2 pages, maybe a page and a half, page 73 to 75. You should also read about the cell walls, page 80 to 83, simple stains on page 65, differential stains on page 65 to 67. That includes the gram stain and the acid-fast stain. You should also read about special stains, page 67 to 68, so there's 1 page, and the negative staining for capsules, that's page 67, and then also page 75 to 77. The endospore stain, page 68, and page 92 to 94, and then the flagella stain, page 68, and page 77 to 78. Any questions about what we're going to do today? All right. So there are some video clips in today's lab, as well as a virtual lab. Your learning objectives for today: 1, be able to prepare bacterial smears for staining. 3, understand the principles and procedures used when performing a simple stain, a differential stain, and a special stain. 3, interpret the results for a simple stain, a differential stain, and special stains. And then 4, define the following terms. What's a simple stain, a differential stain, a special stain? What's a coccus? What's a bacillus? What's a sporillum? What is peptidoglycan? What's a gram-positive cell? What's a gram-negative cell? What is the acid-fast stain? What is a primary stain? What is a mordant? What is a decolorizer? What is a counter stain? What is the cell capsule? What is a cell endospore? And what is a flagella? Any question about any of that? Okay. Bacteria are hard to see because they are essentially transparent. Therefore, it is necessary to stain them before they can be observed, at least well, under the microscope. When preparing a bacteria smear, we add a drop of water, usually sterile water, placed on a clean slide, and then you add a little bit of bacteria, very little, using an inoculating loop. The bacteria are then mixed in the drop of water, and smear it over the surface of the slide. We only add a little bit of bacteria because we want our bacterial smear to be no thicker than a single layer of cells thick on the slide. You then allow the smear to air dry, and then the organisms are fixed under the slide by gentle heating. We usually call that heat fixing. If you do not allow the bacteria to air dry and heat fix on the slide, the bacteria will not stick to the glass and they will wash off the slide in the staining procedure. Any question about any of that? All right. A simple stain uses one dye to stain bacterial cells. You notice how I got the word basic crossed out because it doesn't matter if you use a basic dye or a acidic dye. It's one dye, and that makes it a simple stain. With a simple stain, you can only tell the bacteria shape and the arrangement. You cannot tell anything else because with a simple stain, all the cells will stain the same color. Anything else? Any other questions? Or whatever. Are there any questions? A differential staining technique typically uses two or more dyes, doesn't require that, but typically has two dyes, and provides additional information about the cell. The differential stain can allow you to separate two species visually by their different staining characteristics. Any question about a differential stain? The most commonly used differential stain is the Gram stain in microbiology. Lastly, we have special stains, which stain a particular bacterial structure, such as the cell capsule, the endospores of a cell, or the flagella. So the special stain is staining a special part of the cell structure. In this figure, we're seeing different cocci and some of the different growth patterns or the growth characteristics that they can take. This is usually called the cellular growth pattern or the cellular growth arrangement or the cellular arrangement. And here you can see streptococci. That's cocci lined up in a string. You can have singlet cocci, meaning a single cocci, and that's not shown here. You can have diplococci, and that's two cocci stuck together, shown here. You can have tetrads, and that's shown there. And then you can have staphylococci, and that's shown here. Staphylococci literally means ball-shaped cells, meaning spherical-shaped cells, growing in a cluster like a grape cluster. And that, if you put a stem on that, that sort of looks like a grape cluster. Any question about any of that? All right, in this picture here, we have different shapes of cells. We have a spirilla bacteria here, and that's a S-type shape. We have bacilli here, and here we have coccobacilli. If you have bacilli with a rounded edge, and then you make this cell smaller and smaller, you eventually get a coccobacilli. Coccobacilli are in between the shape of a cocci and the shape of a bacilli, and that's why we call it a coccobacilli. There are a few species which are coccobacilli in shape. The majority of bacteria are either cocci, bacilli, or spirilla. But there are a few that are coccobacilli, and it's in between shape, either a shortened bacilli that almost looks like a cocci, or an elongated cockeye, and that's coccobacilli. Most species are monomorphic, meaning they have one shape, but there are some species which are pleomorphic. A pleomorphic species has many different shapes that the cell takes. Most of the species, if not all of them, at least most of the species which are pleomorphic are bacteria that lack a cell wall. And that's why they're pleomorphic. They don't have the cell wall to give the shape to the cell. Any question about any of that? All right, that's it for the simple stains. Let's talk about the differential stains. There are two. There's the Gram stain and the acid-fast stain, which I think is down next. Wow, it's way down here. It's way down. All right. Those are the two most common differential stains. The Gram stain being by far the most common differential stain used in microbiology. The Gram stain was invented in 1884 by Dr. Hans Christian Gram. You don't need to know the year he invented it, but you should know the Gram stain was invented by Dr. Gram. It's named after him. And so when you say Gram stain, you should capitalize that G because we always capitalize. What the heck happened to Gram there? I would say I made a mistake on this and put in something where Gram used to be. Dr. Gram invented the stain. The stain is named after him, and we always capitalize somebody's last name. So Gram stain should be capitalized in the G. The Gram stain distinguishes bacteria in two large group of cells. Let me state that Hans Christian Gram used the Gram stain as a differential stain to differentiate in his patients between the patient's cell and the bacteria cells. So what Dr. Gram was using it for was to stain the bacterial infection cells a dark purple, and then the patient's own cells were staining pink. And he was using it as a differential stain, but microbiologists started using the Gram stain, and they discovered that it separated bacteria into two large categories of cell. The gram-positive cells, which stain purple, and then the gram-negative cells, which stain pinkish. The gram-positive cells retain the crystal violet, which is the primary dye, the first dye used in the stain. And so these cells remain purple, and we call them gram-positive. The gram-negative cells lose the crystal violet and then pick up the second stain, which is called the counter stain, and they will appear pink. Any question about any of that? And here we can see some cells, gram-negative bacillus there, gram-negative spirillum there, a gram-positive bacillus there. I thought there was a gram-positive cocci down there, but I'm not seeing it. I wonder if this got moved. Yep, it did get moved. Sorry. Let me move that a little further out too. That's a little better. Probably that got moved. Yep, that's not working right. That didn't work. You go back on that one. Let me save that because that's about as good as I'm going to get it. So there's some gram-positive cocci right there and there, which isn't shown here. All right, any question about any of that? The gram stain is dependent upon the cell wall. The bacteria, which are gram positive, have a different cell wall than the bacteria which are gram negative. Shrink this down a little. So here we're looking at the gram positive cells. They do have a plasma membrane, and then they have a very thick peptidoglycan layer. It's between about 30 layers of peptidoglycan and over 100 layers, depending on the species. The gram-negative cells do have a peptidoglycan layer, but it's much thinner. The peptidoglycan layer is between 3 and 20 layers of peptidoglycan. Let me see if I can blow that up, and then you can see that a little better. So here we can see there's a layer of peptidoglycan, there's another layer, there's a layer, there's one layer, there's a layer, there's a layer, there's a layer, there's a layer, and there's probably 2 more layers there. The point is the gram-negative cells have a thinner layer of peptidoglycan, and the crystal violet iodine complex washes out of this thinner layer of peptidoglycan. The gram-positive cells have a thicker layer of peptidoglycan, and the crystal violet iodine complex does not wash out of the peptidoglycan layer, at least if the gram stain is done correctly. We'll talk about the difference in the gram-negative and the gram-positive cell walls later, but you can note that the gram-negative cells have a lipid layer, it's actually a bilayer, outside the peptidoglycan layer, and that lipid layer is absent on the gram-positive cells. Any question about any of that? All right, after bacteria have been stained with the crystal violet, Graham's iodine is used as a mordant. A mordant is something that chemically combines with the first stain, in this case the crystal violet, to make a crystal violet iodine complex. The crystal violet iodine complex is larger than the crystal violet and iodine alone, and that makes the complex less likely to wash out of the gram-positive cells. And then the next step of the gram stain is the alcohol acetone wash. It's used as a decolorizer. It's applied briefly to remove excess crystal violet and iodine. The gram-negative cells quickly decolorize. The gram-positive cells do not quickly decolorize. And the difference is the thin layer of peptidoglycan allows the crystal violet iodine complex to wash out. The thick layer of peptidoglycan does not allow the crystal violet iodine complex to wash out easily. It can wash out if you were to use the decolorizer for too long. It can wash out of the gram-positive cells. And then again, if you did not use the decolorizer long enough, you would not wash at least all of the crystal violet iodine complex out of the gram-negative cells. So the timing of the decolorizer is critical. And then after the decolorization, the gram-positive cells retain the purple color, but the gram-negative cells are clear. And that's because the crystal violet gram iodine has been washed out. We then apply a counterstain to add color to the gram-negative cells. Safranin is the counterstain, and it is reddish or pinkish in color. A counterstain is just the second stain you use after the primary stain. All right, this is something I need to talk about, and that is some cells do not take up the gram stain very well. And they either stain do not stain with the gram stain, or they stain poorly, or they may be gram variable. Like this is one species of bacteria, and you can note some cells are staining gram negative, other cells are staining gram positive. Why are they staining gram variable? Well, there's actually two reasons, and that is this species is a species of Mycobacterium. It has a lot of mycolic acid in the cell wall. That mycolic acid prevents water from crossing the cell wall. The stains of the gram are aqueous stains, so they are in water. And that is why the family Mycobacteria do not stain well with the gram stain. They actually do, if you do the gram stain correctly, at least all the species that I've looked at do stain gram positive, but they stain lighter than most other gram positive cells. And that's just because the cells resist taking up the stain. So that's one reason, and that is the dye is having a hard time getting across the cell wall and staining the cells. And then the second reason is because the student who made this, or maybe the instructor who made this, did not use freshly grown cells. In the gram stain, you should be performing the gram stain on cells that are no older than 2 days old. And if the cells are older than 2 days old, you shouldn't use the cells to do the gram stain because the gram stain may stain incorrectly. If the cells are older than 2 days old, you should streak it out and then within 2 days, stain the cells that you grew up. So generally a day or 2 days later, stain the cells. Any question about that part of the gram stain? All right, the second reason why these are showing variable, I've now blown it up. Do you notice something different between this gram-negative cell and that gram-negative cell and these two gram-positive cells? Notice something different. Anyone? Anyone notice a difference? There's
The edges are more clear.
Yeah, the edges of the gram-positive cells are much more clear. The cell is much more distinct, and the boundaries of the cell are much more distinct. That cell there, it's not even clear where the boundary is, especially in that region there, and the same with that cell there. This cell actually looks like a ghost, and that's probably because it's dead, and the cell is then you're looking through it even though it's stained because it's ghostly. This cell here actually looks like it's got a break in it, and that's probably because it's a dead cell. In fact, it may have two breaks in it. There may be another break there. So these gram-negative cells are old, dead cells. Maybe there's a few that are dying, but they're dead cells, and they're not taking up the dye correctly. And on the other hand, the gram-positive cells, the purple cells, are viable cells, and that's why the cell has a distinct shape, distinct boundary, because the cell is alive. And what happened here was whenever I've seen the gram variable result, it's always been species of mycobacteria. And I've asked the student, how old are these cells? And every student who ever got a gram variable result, they told me, oh, these cells are older than 2 weeks old. You should not be using the gram stain if your cells are older than 2 days old. You should be doing the gram stain on cells that are 2 days old or younger. Otherwise, the gram stain may not stain correctly. So the reason why there's gram variable is because the gram-negative cells are probably dead cells, and then the gram-positive cells are probably viable cells. Any question about any of that? And if they'd done the stain on cells 2 days old or less, they wouldn't have all these dead cells in there. All right, tips to prevent getting gram variable staining bacteria. Avoid using old cells. Prepare a thin smear. If you have a thick bacterial smear, it's really hard for the crystal violet to get all the way down to the bottom layer of cells. And it's also hard if it's a gram-negative cell for that crystal violet to wash all the way out of the layer of cells when you have several layers of cells. So you should have a thin smear, bacterial smear. You should heat fix the bacteria onto the slide only after the smear has air dried. If you heat fix the bacteria when there's water on the slide, it will cause the water to boil, and that will create holes in the cell wall. And then the dye can get in the cell when it shouldn't and can get out of the cell when it shouldn't. You should also avoid heating the cells during the heat fixation. If you overheat the cells, it will make the cells look like they're melted, and they will disrupt the cell wall. So the gram stain may not stain the cells correctly. You should also use fresh staining reagents. Do not over decolorize or under decolorize. If you under decolorize, the crystal violet iodine complex will not be totally washed out of the gram-negative cells. And then if you over decolorize, you'll start washing the crystal violet iodine complex out of the gram-positive cells. The best way to determine how much decolorization to add, I don't know if you guys can see this. Let me go and turn on my light behind me, and hopefully you'll be able to see what I'm talking about, because I don't think you can see this against my face. Can you even see my face?
Yes.
Okay, good. Because the lighting is really poor at the moment. I was using the sun earlier, and it's gotten dark now.
Well, now you're gone.
Well,
Oh.
yeah, I went to go get the light switch.
Copy.
All right, now I'm back. Can you see my face?
Yeah.
Okay, so here's the slide. I'm going to pretend this is the slide. What you should do when you're adding the decolorizing agent is you put it on the slide and you just move the droplet back and forth until you see the blue stop running out of the cells. Once the blue stops running, you should stop adding the decolorizer. And that should happen in about 5 seconds. So that's all the time you need to put the decolorizer on. And like I said, the best way to do it is simply to add it until the blue stops running. All right, we'll talk about the gram stain a little bit later in this lab. Are there any questions so far? If not, let's talk about the acid-fast stain. Some bacteria, mainly the family Mycobacterium and the family Nocardia, have a waxy substance called mycolic acid in their cell walls. This waxy substance resists the acid alcohol decolorization, and so we call these cells acid fast positive bacteria. Most other bacteria, in fact, every other family I can think of, other than Mycobacterium and Nocardia, do not have mycolic acid in their cell wall. And with the acid fast stain, they will stain negative. And these we call acid fast negative bacteria. Any question about any of that? All right. Carbol fusion is the primary stain in the acid fast stain. And there are two of them. You don't need to know that there are two of them and what the difference is, but there are two acid-fast stains, and both of them use carbol fuchsin as the primary stain. Heat can be used as the mordant to help drive the carbol fusion into the acid-fast cells, acid-fast positive cells. Initially, all cell types will stain with carbol fusion, the primary stain, which is a cherry red or pinkish color. So both the acid-fast positive cells and the acid-fast negative cells will take up the carbol fusion. We then decolorize the cells with an acid alcohol decolorization agent, which will pull the carbol fusion out of the acid-fast negative cells. So the acid-fast positive cells will retain the carbol fusion and stain pinkish. And then after the decolorization step, the cells are clear. So we add a basic dye as the counter stain. It's usually methylene blue, which is bluish in color, or brilliant green, which is green in color. And then the acid-fast negative cells will stain either blue, if using methylene blue, or green, if using brilliant green, as the counter stain. So here's an acid-fast stain, and we have two cell types here. The green cells are the acid-fast bacteria. Boy, this got moved. See if I can move that. I can't even move that. Well, that's great. Lost it now. I can move it up and down, but I can't move it side to side. Oh well, I'm just going to leave it there and state that the acid-fast positive bacteria are the pink cells, and the acid-fast negative bacteria are the, in this case, green cells. Let me see if I can tab it. That works. Oops. I'm making it smaller though. Yeah, it's not working. I don't know why I'm not able to move that. Obviously, I accidentally moved it and I'm not sure what the problem is. All right. Any question about any of that? So that's the differential stains. There's two of them. Let's talk about the special stains. There's three we're going to talk about. Remember, a special stain is a stain to stain a special structure in the cell. The first special stain we're going to talk about is one that we call the capsule stain or the negative stain. Some cells produce glycocalyx, which, if the glycocalyx is firmly attached to the cell wall and is visible under the light microscope, we call it a capsule. If the glycocalyx is not firmly attached to the cell wall, is mostly made up of water, is not visible under the light microscope, we call it a slime layer. Any question about any of that? All right, a production of a cell capsule can be associated with an organism's virulence or its pathogenicity. And this is because the capsule protects the cell from being phagocytized by a white blood cell or an immune system cell. Capsules also make the colonies look a little different. And as you can see, these colonies have a mucoid appearance, and that's because the cells are, did I say capsule? These colonies have a mucoid appearance because the cells have a cell capsule around them. Capsules are not easily observed using conventional staining methods. Like with the gram stain, you almost never see a capsule. And that's because the capsule washes off in the gram stain procedure. Therefore, we use a gentle staining technique called the negative stain or the capsule stain, which is employed to show the presence of a capsule. For both of these stains, like this India ink stain, which is a negative stain, what we do is we put the cells on the slide. These are actually live cells, and then we in broth or water, whatever, and then we add the India ink, which is dark, and it simply stains the background of the cell. India ink also lightly stains the cell, sort of like a light gray, and then the black is the background of the cell. The capsule then shows up as a clear region around the cell. This one here is also a capsule stain. This one probably uses two different dyes, one to stain the cell, and then one to stain the background of the cell. And with that, once again, you can see the capsule as this clear region around the cell. Did I mention to you why it's called the negative stain? It comes from the old Polaroids, where when you looked at the negative of a Polaroid, where the face would be light, on the negative it would be dark, and then when the face would be dark, on the negative it would be light. All right, any question about the capsule stain, also called the negative stain? All right, let's move on to the endospore stain, the third special stain we need to talk about. This stain is for staining the cell's endospores. Endospores are a metabolically inactive, hardy structure that are produced by some bacteria as a defensive strategy to survive unfavorable environmental conditions. When the environment becomes favorable again, the endospore will germinate and the bacteria will return to their normal metabolically active vegetative state, which will go through metabolism and reproduction and reproduce. But the endospore is essentially metabolically inactive and does not reproduce. The endospore is a survival structure. It is not a reproductive structure. Most species of bacteria that produce endospores are in one of two genera, I guess if it's plural. And I sometimes call them families because each genera has a family, and that is the family Bacillus and the family Clostridium. There are two species of bacteria that make endospores that don't belong in these families, but each species, there's only one member of each family, so we're not going to talk about it. And most of the important bacteria that we're going to talk about in this lab that make endospore are in the family Bacillus or the family Clostridium. And the important bacteria we're looking at is here. These are the pathogenic endospore forming gram-positive bacilli, and all of them are either Bacillus or Clostridium, in the family Bacillus or the family Clostridium. You will need this table to answer some of the questions in the lab worksheet today. Any question about any of that? All right. The Schaefer-Fulton endospore stain, sometimes just called the endospore stain, uses malachite green as the primary stain. It will stain all cells, both vegetative and endospores, green. Heat is then applied as the mordant to help the malachite green get inside the endospore. The cells are then decolorized with just water, which will remove the malachite green from the vegetative cells, but not from the endospores. Safranin is then applied as a counterstain to stain the vegetative cells a color, in which case it'll be pink. So vegetative cells will appear pink and the endospores will appear dark green. This is so much nicer than my first lab. In my first lab, I had Grammarly having an icon, and I couldn't get to this button to enlarge things or shrink things. These green things here are endospores. They are normally made inside a parent cell. And right there you can see the end of the parent cell and the other end of the parent cell with an endospore in between. When the parent cell dies, the endospore will be released. And so those endospores have been released. These endospores are still inside their parent cell. Any question about any of that? So endospores will appear dark green and the vegetative parent cells will be pinkish in color. All right, the flagella stain is a stain that we use to stain flagella. Flagella are long thread-like appendages of cells that allow the cell to move. If the cell can move, if it's a bacteria or an archaea, it has at least one flagellum. It may have many flagella, but it has at least one if it can move or swim. If it cannot move, the cell does not have, a bacteria does not have flagella. That's not true of eukaryote cells. Eukaryote cells may move because of three structures, and that is a eukaryote cell can move because it has a flagella, but a eukaryote can move if it has pseudopods, and a eukaryote cell can also move if it has cilia. To my knowledge, a eukaryote cell only has one of these three, but if it can move, it has one of those three. Bacteria only have one structure for movement, and that is a flagellum, or flagella if there's more than one. Flagella are composed of the protein flagellin, and the number and distribution of flagella on bacteria cells can be useful in helping identify and classifying the species of bacteria. You can have flagella which are monotrichous, and that means there's only one flagella on one end of the bacteria. You can have bacteria which are lophotrichous, and there are multiple flagella on one end of the bacteria that are lophotrichous. And then you can have amphitrichous bacteria that has multiple bacteria on both ends of the bacteria. And then you can have peritrichous bacteria, and these bacteria have flagella all over the cell. Let me show the picture here. Here we have the picture there. Let me blow that up just a little. Here, this is one species of bacteria. You can see that this bacteria is Peritrichus. That one looks like there's no flagella, so that one's no flagella. This one looks like it has one on one end, so that would be Monotrichus. Let me see if I can find a Lophotrichus. I think there's only one. Oh, right there. There's a Lophotrichus. Multiple flagella on one end. And then let me see if I can find an Amphitrichus. Multiple on two ends. I think there's only one in here too. I'm not seeing the Amphitrichus and I don't remember where it is. Maybe that one, but I'm not certain. So, what is the flagellar arrangement of these cells? Is it monotrichous? Is it no flagella from this one? Is it peritrichous from these three? Or is it something in between? Or is it all of the above? Anyone have a guess? Oh, there's the Grammarly icon. I'm glad it's moved there and not there. Anyone have a guess? Well, think about it, because I'm not going to tell you unless you guess. All right. Flagella are too thin to be seen using standard staining techniques. You need a special stain in order to see the flagella. One special stain, which is a flagella stain, simply coats the flagella many times. And after you have many coatings on the flagella, you can then view them under the light microscope. But there are other flagella stains, and I'm not sure how all of them work. Any guess on what the flagella arrangement for this is? You should guess because you're going to get that question in the worksheet, or at least a similar question. All right, no one's going to guess, so I'm moving on. You need to know the terms to this lab. You don't need to know about the references. In this lab, you're not going to be physically performing any of these activities, but you must understand how to prepare bacterial smears before staining, and then the principles and procedures used in the various staining techniques, and be able to interpret those results from the microscope. And you must be able to understand the differences between the simple stain, the differential stain, and the special stains. Anyone doing a guess on the question I asked?
Can you repeat the question?
What is the flagellar arrangement of this species? It's only one species for these cells. Is it peritrichous, amphotrichous, lophotrichous, monotrichous, or that one has no flagella? Or is it all of the above?
Peritrichus?
You are correct. What you do is when you're determining the number of flagella, you take the highest order you can find in the picture, because once a cell divides, it'll initially have no flagella. And then it will grow one flagella on one end, and it'll be monotrichous. And then with more time, it'll grow more flagella on one end and be lophotrichous. And then it'll grow flagella on both ends if it's amphitrichous. And then if it is peritrichous, it'll grow flagella all over the cell. So you name it by the highest order of flagella you see on the slide, and you understand that the other ones are growing their flagella, and that if given enough time, if the cell survives, it'll eventually become peritrichous. Okay? But there are different species. This one is obviously peritrichous. There are other species where the highest order is amphitrichous. There are other species where the highest order is lophotrichous. And there are some species where there's only one flagella on the cell. And those cells, you'll only find cells with no flagella or monotrichous flagella. And then there's many species of bacteria which have no flagella, and that's because these cells do not move. They don't ever produce a flagella. All right. I think that's it for the lab module. Let me go down here. I think everything here is in the exercises. So let me go to the exercises. So you should answer the following. In procedure 1, we're preparing a bacterial smear for staining, but we aren't doing the staining procedure. We're only doing the smear or bacterial smear procedure. And I'm trying to emphasize that a bacterial smear is separate and distinct procedure than the stain. A bacterial smear does not have to be stained. Like somebody makes a bacterial smear and then they look at it directly under the microscope, having no stain, or they make a bacterial smear for some other reason, like maybe they're going to run a test on it. So bacterial smear is separate from staining and independent. Okay? And then we have the staining procedure, which usually uses a bacterial smear first, and then you do the staining procedure. But you don't have to make a bacterial smear to do the staining procedure. Like, for example, you use a wet mount and then you stain the wet mount. You're not ever making the bacterial smear. I imagine you could put cells on a slide. I've never done this, and not do the bacterial smear and then directly stain the cells. You'd probably only want to do that if you only had a few cells to stain, probably a larger cell than a bacteria too. But you could do the staining procedure without doing a bacterial smear. And certainly you can make a bacterial smear without doing the staining procedure. So I'm trying to emphasize these are two separate procedures because students get them confused and they start talking about staining when they should be talking about bacterial smear, and they could talk about a bacterial smear when they should be talking about a stain. All right, so view the video clips. We have a video on preparing a bacterial smear and then air drying and heat fixing it. And then review the steps for making a bacterial smear. First, you put a drop of water, usually sterile water, on the slide. You usually want sterile water so you don't add any bacteria from the water. And you use a clean slide. And then you spread that drop of water after mixing. You mix the bacteria and the drop of water, and then spread that drop of cells and water all over the slide. Part of the reason is, besides to spread it out, you also want to make that drop of water thinner so it can evaporate quicker, because you do need to let the slide air dry, and then you heat fix it. Heat fixing sticks the cells, air drying does a little bit, but heat fixing sticks the cells, the bacteria, to the glass of the slide. It also has the advantage that it tends to kill many bacteria, like E. coli typically is killed by the heat fixing step. But not all species will be killed by the heat fixing step. For example, if you have a bacteria that makes endospores, the endospores are going to survive the heat fixing. You then let the slide cool. Usually that's just for a few seconds. And then the smear is now ready for use, such as staining the smear. But remember, staining is a separate procedure from making the smear. And then in procedure 1, in question 3 or item 3, what are 4 common errors when preparing a bacterial smear for staining? So give me 4 common errors, and that will be something you'll be graded on. If you don't answer that question, some students don't, you'll get a minus 0.4 on this lab. Procedure 2, simple stain. View the following video clip of a simple stain and then review the procedures. With a simple stain, you're adding 1 dye, either a basic dye or a negative dye. And usually you're adding it to a bacterial smear. You put the dye on for about 1 minute, and then you wash it off. You shake off the excess water and blot the slide dry. What you do with blotting is you grab some blotting paper and you rip off the blotting paper. Yeah, that's not going to work well. Let's go with this one. And then what I like to do is I fold the blotting paper and then stick the slide. I'm pretending this is the slide and this is the blotting paper. Fold the blotting paper, and then you just press down the blotting paper on the slide. You do not move the blotting paper against the slide this way, if you can see it. You do not move the slide this way against the blotting paper, because if you do that movement, you will pull off the cells onto the blotting paper. And all you want to do is block the slide so that you pull off the majority of the water. The blotting like that will remove enough of the water that you can then view the slide under the microscope immediately, including if you're going to use oil. You remove enough of the water from the slide so you can even use oil on the slide, and then view it under the microscope. All right. In 3, there are 2 smears using a simple stain. Tells you what power we're using, what objective lens or something here. The oil objective lens, there it is. And then answer the question A. That's procedure 2.3a. What is the cellular morphology? That's the shape of the bacteria, not the cellular growth arrangement of E. coli and Staphylococcus aureus. You can blow it up if you want. Oh my gosh, I got that in the way now. I'm not going to blow it up. I'm going to warn you that Staphylococcus E. coli is not spirillum or a spiral. That right there is probably 3 cells stuck together. That right there is 2 cells stuck together. So give me the shape of the E. coli and give me the shape of the Staphylococcus aureus. And then for procedure 2, question 3b, what's the total magnification? So give me the magnification. For procedure 3, we're looking at the gram stain. We have a video going through the gram stain, so watch that video. And then you can practice performing the gram stain by doing the virtual lab. Let me open that. It may take a little while to download. There we go. I'm going to shut that down. I'm going to move very quickly through this. Why is that not showing? There we go. I'm going to move very quickly through this. You can read through it. I'm going to move to the part of the gram stain. Actually, there's one part I will talk about before the gram stain, but Whenever you need to do something, it'll have an arrow, and that's telling you start from this, take your mouse, click on what you're pointing at, and then bring it down in the direction of the arrow. And that tells you how to, in this case, light the Bunsen burner by taking the mouse and bringing the flint striker down to light the Bunsen burner. And it also tells you how to work this. Oh, it doesn't tell you to click on that. You need to click on that. I don't know if you, yeah, I guess you can click on there too. Let's just continue. 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 Like I said, you can go through it. In fact, you should go through it. It's part of the lab. I'm just moving to the Grams tank because I want to talk about that. And that's actually a little bit wrong. It showed you they were heat fixing the slide when there was water still on the slide. You don't want to do that because that'll cause the water on the slide to evaporate, and that will cause holes in the cell wall that may disrupt the staining of the gram stain. All right, so this is the start of the gram stain. Let me go ahead and turn my
the slide off under
volume.
slow running water in between each chemical. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample. Let that sit for 60 seconds. Now rinse it under slow running water. Now use the bottle of Grams iodine. Grams iodine will fix the dye to the cell walls of the bacteria and make it permanent.
Actually, it's not entirely true that it's permanent. You can wash the crystal violet iodine complex out of the cell wall of a gram-positive cell if you apply the decolorizing agent for too long.
Squeeze several drops onto the sample, enough to cover it completely. Let that sit for 60 seconds. Now, rinse it under slow running water. Now we are going to use the alcohol solution. This will wash off the excess dye, leaving only the dye fixed to the bacteria.
This is the decolorizing step. In this virtual lab, they're using 95% alcohol as the decolorizing agent. In the lecture, as well as in our lab, we tend to use acetone alcohol or alcohol acetone, which is a stronger decolorizer. So the 95% alcohol is left on the slide for a longer period of time than the alcohol acetone. So we usually warn you, you shouldn't use the acetone alcohol for more than about 5 seconds. It's best to actually watch when you're applying it and stop applying it when the blue stops running. But the 95% alcohol is a weaker decolorizing agent, and it needs to be on the cells longer. Oops.
Use the alcohol dropper to cover the sample in alcohol. Let the alcohol sit for 30 seconds. Time is very important in this step. Don't let the alcohol sit for more than 30 seconds.
And another thing is you have to have the alcohol sit for very close to 30 seconds, because if you were to let it sit for much less, like 10 seconds or less, you would not pull out the crystal violet iodine complex from all of the gram-negative cells. So time is important, and it does depend a little bit on which decolorizing agent you use. In this virtual lab, remember, we're using 95% alcohol. It's a weaker decolorizing agent than the alcohol acetone or acetone alcohol decolorizer that we use in the lab here, as well as in the lecture. All right, that's it that I wanted to talk about. You can go through the rest of the gram stain here. You should go through the virtual lab. It is a part of the lab. And then go through the steps of the gram stain. First you apply, well, first you have the bacterial smear. You have to make a bacterial smear, let it air dry and heat fix, and then it's ready for the gram stain. First, you add the crystal violet to the bacterial smear. You let it sit for a minute, and the crystal violet will change the color of all the cells, both gram negative and gram positive, to a bluish purple color. You then wash that off and then add the grams iodine. Iodine will complex with the crystal violet, making a larger molecule than either the crystal violet or the iodine alone. It will also change the color a little bit. So instead of a bluish purple color, it'll be a purple color. And it may, although this is hard to see, it generally will intensify the color so that this would be a little bit more intense color than that one. And that's not shown. Another thing that's not shown is the slight change in color. You then rinse that off after letting it sit for a minute, and then you use the decolorizing step. Here we're using alcohol acetone as the decolorizer, and it should only be on for about 5 seconds. The best way to apply it is to actually visually look at the slide when you're applying it and just move the what is that called? The eyedropper back and forth. And then when the decolorizer stops running blue down, stop adding the decolorizer. Then you should immediately wash the decolorizer off the slide. The reason being is the decolorizer, even if it's only trace amounts, it's given time, and with more time, it'll start to pull the crystal violet iodine complex out of the gram positive cells. So you should then immediately wash the cells. If it's done correctly, the gram stain, the gram positive cells will retain the crystal violet iodine complex, but it will be pulled out of the gram negative cells. So the gram negative cells here will be clear. We then apply safranin as the counter stain, and it will stain the gram negative cells pinkish in color. Any question about any of that? All right, so where did I lose it? There it is. Below are 2 smears viewed using the oil objective lens that were gram stained. Complete the following table. Give me the gram reaction if it's gram positive or gram negative for E. coli. And this is probably blown out. Let's go to 100% and see if that's better. That's still a little bit off. That's the E. coli. That's the Staphylococcus aureus. So give me the gram positive or gram negative for the E. coli, and then give me the cell morphology, Coccus or Bacillus. For Staphylococcus aureus, give me the gram reaction, and then the cell morphology. In procedure 3, also answer question A, B, C, D, E, and F. A, ask, what is the color, pink, purple, or colorless, in the gram stain after the Staphylococcus aureus has been given the primary stain? So give me the color. And then what is the color of E. coli after the primary stain is added in the gram stain? Pink, purple, or colorless. And then C, what's the color of Staphylococcus aureus after the alcohol acetone step? What is the D, 3D, what's the E. coli color after the alcohol acetone step? 3E, what's the color of Staphylococcus aureus after the counter stain is added? And then 3F, what's the color of E. coli after the counter stain is added? All right, any question about the gram stain? In procedure 4, we're doing the acid-fast bacteria stain. Here's a video clip on the acid-fast bacteria stain. We're going to do this step using the modified Kenyon acid-fast bacteria stain. You add carbol fusion to the cells, and both the acid-fast positive cells and the acid-fast negative cells will both stain pinkish or reddish in color. You then add the decolorizing agent, which in this case is an acid alcohol wash. Let me blow that up a little so I can see that. Let's see if I get it. Nope. There we go. Well, didn't quite get it. There, I got it. After adding the decolorizer, the acid-fast negative cells will lose their carbol fusion and be clear. And then the acid-fast positive bacteria will continue to be pink from the carbol fusion. You then apply the counter stain, which in this case is brilliant green, and the acid-fast negative cells will stain greenish, but the acid-fast positive cells will stain pinkish. For procedure 4, you have to answer the questions related to the arrows, and with each arrow, it says, give me the, list the arrows that are acid-fast positive or acid-fast negative. Supply an answer for each arrow. You can write it here, or you can write it up above the picture or below the picture. But you do need to answer all 4 of the arrows for full credit. If you do not answer this question, you'll get a -4. Procedure 5 is looking at special stains. View the video clip on an endospore stain and a video clip on the negative stain for a cell capsule. And then below are 3 bacterial smears that are stained and being observed at 1,000 times total magnification. Figure A goes to question A. Figure B goes to question B down below. And then figure C goes to question C, which is actually shown right there. So in question A1, it says this is the Schaefer-Fulton stain, which uses which stain as the primary stain? So answer which stain. And it ultimately dyes which cell structure produced by some bacteria to aid in their survival. So name the cell structure in, I guess that would be A2. It's A1, A2. And then A3, what are some examples of pathogenic endospore producing bacteria? You can get those examples in the lab module. For procedure 5B, referring to figure 5B, this is what stain that coats long whip-like appendages which aid in motility. So name the stain in 5B. And then you can look at the flagella arrangement here and name the flagella arrangement. And then in 5B3, give me some examples of clinically significant bacteria that produce a flagella. And I forgot to mention the word is examples, plural, so you must give at least 2 examples. And the same with the, let's see, that would be a procedure 5A3. Some examples of pathogenic endospore producing bacteria. You must give me at least 2, because if you give me 1, that's not examples. It is an example. And then procedure 5C, name the stain. This is a stain that uses India ink or nigrosin and can be used to detect the presence of a cell structure which is located exterior to the cell wall and prevents phagocytosis. So name the stain shown in figure C here. What the heck is that? And then give the name of the structure that we're seeing, which is located exterior to the cell wall and prevents phagocytosis. So answer C1, the name of the stain, and C2, the name of the structure that's being stained by the special stain. And then C3, name some pathogenic microorganisms that produce capsules. And once again, you must have 2 because it says some microorganisms. Any question about any of those questions? And then come down to the case study. Case study 1 is a 45-year-old female experiencing some respiratory issues. Based on her clinical presentation, the doctor orders a chest x-ray shown here, and then they do a sputum from the lady, and then an acid-fast bacterial culture. In addition to the culture, the acid-fast bacteria smear is made on the sputum, and then it's stained. And based on the findings below, what is the probable diagnosis? You must name the species which is causing the infection. So if you give me the disease, you only get partial credit. For full credit, you must name the species which is causing the infection. Any questions about that? All right, let me warn you that you can do the case study simply by putting in the information to AI and then asking AI. And I don't know how you would put in the picture, but you could probably describe the picture to AI. You could do that and would be an easy way to get the answer. But if you do that, you will not be prepared for when I give you a case study in the final. And there may be case studies in some of the later quizzes as well, but there certainly are case studies in the final. So you should do the case study on your own. And if you really want to check if your answer is correct, meaning you doubt yourself, you can then put the answer into an AI. But if you put your answer into AI, you will be unprepared for a case study in the final. And you'll probably miss the case study question. Any question about any of that? All right, let me mention again, you must name the species for what's causing this lady's problem. Oh, let me mention a little bit about the x-ray. This x-ray looks unusual. Hey, look at that. I can actually access this. I couldn't for a while because that was blocking it, but now it's moved up there. This is about the color of a normal lung. It should look dark. This area of the lung does not look correct, and this whole area of the lung does not look correct. Okay? And then do case study 2. Case study 2 is a 64-year-old diabetic patient is admitted to the hospital with soft tissue necrosis. And there's the necrosis there. It looks pretty bad. A routine culture and gram stain is ordered. The bacteria grew aerobically, and based on the findings below, as well as the information here, What is the most probable diagnosis? You must name the species which is causing the infection. Once again, you could use AI, but if you do so, I will warn you, you will not be prepared for a case study in the final, and perhaps in a quiz if I have a case study in a quiz. Anyways, there's the patient's foot with the necrosis. There's the Gram stain, and there's the Schaefer-Fulton stain. So find out which bacteria it is, and you can get that from the lab module. Also, if you do AI and you come up with an answer, which is a bacteria species which is not named in the lab module, I'm going to mark it wrong because you're supposed to be using the lab module. You're not supposed to be using AI for the case studies. Any question about any of that? All right. Go ahead and work on the lab. That's it for the lab. I will be here until 8:30 to help you with any questions in the lab, but I will leave once the last student has logged out. So if you have a question, make sure you ask it before all the students log out. Any
Thank you.
questions? All right. Any questions?
I have a question about lab 3, if that's okay. The very last question on there, you have the picture of the microscope, the circle. Are you wanting us to, it doesn't say to, but are you wanting us to like draw something in the circle?
No, you just need to answer the question.
Okay, perfect. Thank you.
All right. I say you must be a student who's taken it from another instructor because that was something that especially Roberto did where you're supposed to draw in there. And I just decided, good heavens, that's way too difficult to do online, as well as the fact that the number of drawings you have to draw in there would be way too many.
Okay, that's what I thought too. I was like, either I'm getting the wrong answer or I'm not supposed to draw it in
You're not
there.
supposed to draw, yeah. All right.
Okay, thanks.
I mean, you can, it's not going to hurt you.